Good afternoon, and welcome to Using NCBI Databases with Tools that Predict Genomic Variant Effects. My name is Ben Busby, and this afternoon, Caitlin Williams, who is an NIH postback in the Genomics Outreach and DB SNPs group, will be presenting the webinar. I will be answering technical and specific questions as the webinar goes on, and Caitlin and I, as well as other NCBI staff, will be answering more substantive questions at the end of the webinar. Also, if you have very specific questions about the tools, we do not maintain nor endorse any of the tools here, so we will likely encourage you to contact the actual tool developers. Additionally, if you are a tool developer and your tool works with NCBI variant data and is not shown here, please send us an email. You can either send me an email at ben.busby@nih.gov, or you can send an email to webinars@ncbi.nlm.nih.gov. So without further ado, I'd like to introduce Caitlin Williams, who will be presenting to you for the next 40 minutes or so.
All right, hi. So as Ben said, I'm Caitlin Williams, and I will be going over this webinar with you today. Here's just a quick overview of what we're going to cover. So we'll start off with an introduction to some of our resources, some variant callers that you might be interested in, and then we'll get into going over using NCBI databases with our tools. So we'll start off with tools that take VCF input, and that'll be Anavar, SNPF, VEP, VAST, and VAT. And then we'll go into a couple of tools that use protein FASTA input, and those are SNAP2, Panther, and Propian. So here's just a little blurb about some of our NCBI variation resources. Some of the more popular ones are DB SNP, and that's a database of small genetic variations that are less than 50 nucleotides in length. DB VAR, that are larger structural genetic variations that are greater than 50 nucleotides in length. dbGaP holds genotypes and phenotypes. ClinVar is human variations with clinical data. There's Variation Viewer, that is a visualization tool that you can also download from. The 1000 Genomes Project, which has variant data along with population information. And BLAST, which is an alignment search tool. If you're running raw data, you'll probably first want to use a variant caller before you use any of these tools, just to get it into VCF format. So these are some of the more popular variant callers. We also have a list of them at this website at NCBI-hackathons/community software tools for NGS. And feel free to add more tools to that. I've been working on that for a few months, so feel free to add them, or you can email me with any suggestions. All right, so now we'll get into some of these tools. Okay, so first off, we'll want to go over where you can get NCBI data. So one of the main places is going to be the NCBI FTP site. And so that's for file transfer protocol, and it's a web page where you can access and download data available from NCBI. This is kind of just a demonstration of how our FTP site is set up. So if you go to the main FTP page at NCBI, you can find links to 1000 Genomes data, BLAST, SNP, and SRA directly on that front page. But if you want to find ClinVar or dbVar data, you'll have to go into the pub directory to find those. So here's an example of what a DB SNP page would look like. So as I said, this holds small genetic variations, and this is an example of a RefSNP report. So this gives some of the information that you would be able to find in some of the DB SNP files, just in kind of a visual view of that. So this is an example for RS328. And as you can see, you can get some of the allele information as well as clinical significance and things like that from this. And also as a side note, if you need to search DB SNP for specific variants, or if you're interested in a type of variant or in some type of specific organism, because DB SNP does hold information from many different organisms, you can go into the SNP search bar. You can type in whatever type of functional class you want. So for this example, it was missense, functional class, and Homo sapiens as the organism, and this will give you a list of all of the missense mutations in DB SNP for Homo sapiens. So that might help if you're looking for specific variants of interest. Okay, so now if you're going to download DB SNP data from the DB SNP FTP site, specifically VCF files, you can go into the main NCBI FTP site, SNP, and then you'll go to organisms. And under organisms, you'll be able to find the human files. And so we have files that are mapped to both GRCH37 and GRCH38. You'll go to that, and then you can click on VCF, and you'll get all of the VCF files that are available for that. And as you can see, there's a few different variations. You can do common and all. You can just download all of them if you'd like. So there's a few different files that you can choose from. Next up is ClinVar. So ClinVar contains human variations with clinical data. And this is the main homepage for ClinVar. And as you can see, there are multiple links, and you can get straight to their FTP page from here. And here's an example of an FTP file that is on the ClinVar website. There's a good bit of information on this page, and from this ClinVar search bar, you can do basically the same thing you did with dbSNP, as well as looking up specific genes, so you can find all the variants that are on one gene or related to one condition, things like that. So, and then for the ClinVar FTP site, you'll just go to the FTP site /pub/ClinVar, and then there are actual links for VCF_GRCH37 or GRCH38, and this will give you a list of all the VCF files, and you can download ones that are specifically just common and clinical, or common and with no medical impact. So it just depends on what you're studying. So then there's the Genome Reference Consortium, which houses all of our reference sequences. So that's GRCH38, GRCH37. And there are reference sequences for human, mouse, and zebrafish. Obviously, for our purposes today, we'll be focusing on the human genome, which is GRCH37, which is also known as HG19 or GRCH38, which can also go by HG38. So when you're going to download the FASTA sequences for GRCH37 or 38, you can either do a download of the full FASTA file, or you can download it by chromosome. So for the full download, you'll go to the FTP site /genome/all, and then that /GCA file, and that will take you to this directory where you can download the full report. If you're going by chromosome, you can do, you'll go to /genomes and then into the H_sapiens file. If you want GRCH37, if you click on this archive link right here, that'll bring up past reference sequences, and you can get GRCH37 through that. So for some of you that might not know what a VCF file is, it's a standardized text format, and it's used for storing large amounts of genomic information, including SNPs, indels, structural variants. So it has header lines that start at the beginning that have information about what is maintained in the file, followed by the column headers, and then the record for each variant follows underneath that. Here's an example of what a VCF header might look like. This is actually one for dbSNP. So as you can see, there's a ton of info at the top about the file itself and what all the tags might mean, followed by that header with the columns, and then the record is beneath that. So before we begin, also, for some people that might not be used to working on the command line, you're going to want to remember to unzip or tar xvfz any files that you do download before using them. So for files that end in .tar.gz, you'll want to do tar xvfz. For gz files, you'll want to gunzip and then the file name. And for .zip files, you want to do unzip and then the file name. And I'd like to remind you again to visit the GitHub site, and feel free to contact us with any suggestions that you may have. Okay, so now we'll start off with our first tool, which is ANNOVAR. And this is pretty popular and pretty widely used, and it's a variant annotator. So for our purposes today, we're using a dbSNP file that is mapped to GRCH38. So we'll want to make sure that you download reference databases that are also in the HG38 or GRCH38 format, or else it won't map correctly when you try and match those up. For ANNOVAR, you can download these additional databases. So you can use this command right here at the top, the Perl annotate_variation.pl, and buildver HG38. And then for this specific one, I chose AVSNP 144, which is actually just their version of dbSNP, ref gene, known gene, 1,000 genomes file, and ClinVar from 2016. If you want a full list of all of the extra databases that they do have, you can look up AVDB list instead of a specific name, and it'll pop out all of them. So then running this for annotation, you'll do Perl table anavar, and then you'll want to put in the file. So ours is that all 2015. It's the whole dbSNP file. Human DB, buildver, the reference sequence that you're running with. So HG38 for this one. What you want the output to be in. So for this one, I just made it anavar.test, just to be pretty general. And then you type in the databases that you downloaded that you want to get more information on, and then you'll want to include that -VCF input since that's what the file is, and it will give you a VCF file in return that has been annotated with all this different information. As you can see, there's a lot of information in that last info column on there. You can gain a lot more information about the specific SNPs in the file you're looking at by doing this. Okay, so next up, we're going to go into SNPF. This is also a very popular VCF annotation tool, and as a side note, this also does work with SIFT and Polyfen, which will give you some variant effect scoring. So when you're downloading SNPF, it will actually come preloaded with GRCH37. So you'll want to go ahead and download GRCH38. They actually provide a pretty easy way to do that. So what you'll need to do is you'll type in this Java xmx4d-jar, SNPF jar databases. And then for this, I just grepped out the GRCH38 because I knew that was what I wanted to download. And then once you find the name of the database you want, you can just type in this last command, snpf.jar download, and it will download that reference sequence that you want. So you don't have to worry about finding it from anywhere else. They have it loaded. Okay, so now once that database is downloaded, you'll run this following command, which is snpf.jar, and so you'll annotate it with grch38, that's that reference sequence. And for this one, I ran it with the common and clinical SNPs file from ClinVar, and then you'll get a VCF result as well as an HTML result. So I will open that up. So you'll get a file that looks something like this. And so as we're going in, this will give you some details about the command you just put in, some variant rate details, so how many variants are on each chromosome, the number of variants by type. And then down here, it'll give you what the impact is. So since we're running common and clinical SNPs, a lot of them came up as being modifiers, what kind of functional class there are. Most of them were missense mutations, as well as more charts giving you the type and the region that they're in, the number of variations, and kind of the same thing, but now a chart. And then if you scroll down some more, there's this one that gives you the number of base changes. So as you can see that there are a lot more that go from a G to an A and a C to a T than perhaps an A to a T. And then if you'll scroll down some more, so what this gives you is the triple nucleotide changes that would obviously lead to an amino acid. So as you can see, there's a lot right here that is the CGG to CAG. So there's a lot of changes in that. And then if you go down a little bit more, it actually gives you just the amino acid changes that might happen. So that can be very helpful for gaining more information, and the graphical view is much prettier. Okay, so now that we've gone over SNPF, next we'll talk about VEP, which is a variant effect predictor, and this is actually an ensemble tool. So the basics for downloading VEP are found directly on their site. They'll just download their zip file, unzip it, and then you'll just go to that directory and then install it. So when you're actually installing VEP, you'll get a prompt for if you want to download any cache files. You'll want to say yes, and you're able to download grch38 with refseq accessions. So that is number 44 right there. And you'll just type in 44 after. It'll ask you which one you want to download. It'll download it for you, and then you will have grch38 with refseq accessions that will work with our data already on the put into their tool. And it's important that you make sure that you're using the right reference sequence, and also that it has the right accessions. If it has the wrong accessions, it might not match up correctly, and then you won't get proper results. So once you have that downloaded, you can go ahead and run the following command on the bottom. So it'll be variant effect predictor, cache, and refseq. So that will call that refseq grch38 file that you downloaded. And then for this one, I focus specifically on chromosome 22 in the common and clinical SNPs, and then type what you want the output to be. And as with SNPF, this will also give you a summary page in HTML form that you're able to open up in a browser. So as you can see, this provides some charts about the variant classes. So on this specific chromosome and this specific file, the majority of these were small nucleotide variations with a smaller amount of deletions. And then it has consequences. So a lot of these were missense. There are a few splice regions, and you can kind of scroll around on these and it'll tell you the percentages of what everything was. And then here's all consequences versus the most severe ones. It'll give you some coding consequences. The same, a lot of them are missense. And then variants by chromosome. For this example, I only looked at chromosome 22. But it'll give you the distribution of these and the positions in the protein. And then if you actually scroll back up to the top, it will actually give you all of this in a text file as well that you can also find on the command line. Okay, next up, we're going to talk about VAST, which is a variant and annotation analysis and search tool. So VAST will actually score the variants and give you a score as to what their functional effects would be. So I actually ran a dbSNP file with VAST. So the first thing that you're going to want to do is do this Perl command. And what this will do is this will remove the genotype information from the dbSNP file. This actually causes some problems when you're running VAST with this information still in the file. So once you've done that, you'll make a new dbSNP VCF from that. And then you'll want to convert it to GVF because that is actually what VAST takes for the annotation step of this. So you'll run the VAST converter, make sure you put the build in. Also, side note for this, we're actually using a dbSNP file that is mapped to grch37 or HG19. That's why the reference for this is different. So then you'll run that, you'll get a GVF file. You'll then want to sort the GVF file on a GFF3. So for this one, I used a GFF3 file that VAST actually has available on their FTP site that you can find under their main website, and then you'll hit data links and you can find it under there. And that made it easier to map with that. And then you'll want to do the same thing for the GVF file. You'll just want both of those sorted, and then you'll be able to go ahead and annotate with it. So the first part is you're going to run the back command for them, and then it's going to be the grch37 FASTA file with that GFF3 file that you got from VAST, and then that dbSNP sorted GVF, and that will give you an annotated GVF from that VAC command. So then from there, you'll want to make this CDR condenser file. So you'll use the VST command that's available in VAST. You'll also want to specify that you're still using the reference of HG19, and that will make that dbSNP CDR file. So that will be the target CDR file that you'll want. So it requires that you have a background CDR file. So this is also something that VAST has available. So if you go to this link right here. This is through the VAST website. So you'll download that, you'll gunzip that file, and then you'll have it available. And then you can do this VAST scoring. So then you'll finally run the VAST command. And then you'll want to make sure for this that you do the ref and nomax allele count commands, because this will basically say that VAST will score variants that are found in both the background and target file. It's set up to only score variants that are only found in the target file, but since we're using a target file and a background file that both have dbSNP data, it basically scores nothing. So, and then you'll get a file with a bunch of scored variants that looks a little bit something like this. So what this is that top one is a name for the variant itself, what chromosome it's on, and then this is a bunch of information about the different permutations of that variant. So the first column is the records, the chromosome and contig name. The second column designates the strand that the feature is on, and the third and subsequent column specifies the structural annotation for the exon. And then there's some more numbers separated by colons there, and those are the start coordinate of the exon, the end coordinate of the exon, the strand of the exon, and the chromosome number of the exon. And then down here, you'll be able to find a raw CLRT score for the specific variant. So what this is it's 2 times the natural log of the composite likelihood minus 2 times the number of variant sites in the feature. Okay, so the last tool that takes VCF input we're going to cover is VAT, or Variant Annotation Tool. Before we start going over VAT, I would like to say that this requires a lot of external dependencies that I personally found a little bit difficult to set up as a beginner, and it also requires that you have permission for sudo commands, which I didn't, so I actually had to have a systems person here set it up for me. So while it's a great tool, if you're more of a beginner or if you don't have sudo commands, it might be better to start with another tool, but we're still going to demonstrate this one. First up, you're going to want to download data to run on VAT. So I followed, VAT had a startup guide. I basically just followed those commands and but updated it with data from GRCH38 because theirs was still GRCH37 data. So I just did newer data from NCBI. So you'll want to download this GenCode annotation file as well as an HG38 2-bit file from UCSC. And then you'll download a file from, so for this one I downloaded 1,000 genomes chromosome 22 phase 3 file to test, and you'll want to download this TBI and the all panel too. Those are just backgrounds. You're mostly just going to work with this VCF file. Once you have all of those, you want to go through some of these commands. So the first 3 are just going to deal with that GenCode annotation filtered GTF that you downloaded, or GTF file that you downloaded. So that first command, which all of these can be found on the VAT website, that will go ahead and filter that. And then once it's filtered, you'll run GenCode 2 interval and interval 2 sequences, and you'll create these GenCode annotation filtered interval and filtered FASTA files. And then when you run this last command, which is basically just testing that 1,000 genomes chromosome 22 file in VCF format that you downloaded, you'll do SNP mapper and then run it with that interval in the FASTA file, and you'll get this genotypes annotated file at the end, and that will look a little something like this, and it has a ton of information in here regarding population and things like that, since that is what 1,000 genomes holds, as well as some information on whether it's a SNP or other things of that nature. Okay, so now that we've gotten through some of the tools that take VCF, we'll go into tools that take FASTA protein input. So one way to find FASTA files is actually on Variation Viewer. So as I said, Variation Viewer is kind of a visualization way to see some of our variants and things like that that are held at NCBI. So for the following examples, I just looked at chromosome COMT, or gene COMT on chromosome 22. So that is right here. So if you click on this, it'll come down with some protein files and other things like that that are all on this gene. And if you right click on any of these, you can do a blast search, or you can download the FASTA for this file. Okay, so that is one way to download the FASTA. And then you can blast it and find the FASTA through that. You can also do, this is an example of protein blast. You can also do blast X, which goes from the nucleotide to the amino acid sequence. So this is an example of what a protein blast page would look like. And if you scroll down, it gives a ton of alignment information, similar proteins that might be in other organisms, a lot of really good stuff. And then you can also go to the protein database on NCBI, search for protein, and this will give you, you can see a graphical view of it here or the FASTA file. So once you have that FASTA file downloaded, you can use some tools. So first up is going to be Snap2. So this is pretty simple. So you're just going to copy and paste the FASTA into the Snap2 entry right there, and then you'll just hit Run Prediction. So once this is done, you'll get something like this. This actually shows the amino acid sequences at the top here, and then the potential amino acid changes are on the side. So as you can see, more blue means it's neutral, whereas more red means there's an effect. Like some of these, so this tryptophan getting changed to basically anything is going to be a higher effect, so versus some of these like leucine. And then it also gives you this chart, so you can jump to a residue position. So we're looking at residue position 52 here, which is a glycine. So when a glycine gets changed to, let's say, another glycine, obviously that's going to be very neutral. So the more negative the number, the more neutral it is. The higher or more positive the number, the greater the effect would be. So for, let's say, if it gets changed to a Y here, there's going to be a greater effect. So you can gain a lot of information about some of the amino acid changes and what that would do to the protein overall from this. Next up, we'll look at Panther, which has a tool that gives a hidden Markov score. So you'll basically do the same thing. You'll enter the FASTA protein sequence, hit submit, and this will give you an alignment of that as well as what that e-value score would be. If it's less than 1 to the -3, no hits are reported. So obviously there are no hits reported on this since it's to the -179. And so then Panther also offers a CSNP search tool. So for this one, you're going to paste in the FASTA sequence followed by the different substitutions that you specifically want to look at. So for this one, I just picked out 4 random ones. So that would be a glycine to a tryptophan at 52, an alanine to a histidine at 117, an isoleucine to a leucine at 222, and a cysteine to isoleucine at 34. So once you type those in, you can hit submit, and you will get scores for like this. So for this P deleterious, the higher the number, the more deleterious it would be. So you can gain some information. So this one would do a lot more damage to a protein than this bottom one, which has a much lower number. Next up, we're going to look at running Provian, which does something pretty similar to what the Panther CSNP tool did. So Provian works directly with NCBI Blast and the NCBI Blast NR databases. If you want some information on running those, you can find those on our webinars site. And so this will compare FASTA files and give possible effects for specific variants. So to run Provian, you will need to download all of NCBI Blast and those NR databases. So you'll use the following command. So this FASTA file is just the FASTA protein file from Blast, and that .var file is just a text file that just contains the variations that we used earlier, just a list of them. So then when you do this, it'll score those variations for you. It's a delta alignment score, and basically what this means is if you have a specific threshold score, if it's, let's say it's negative 2.5, so this would mean that those first two variations would be deleterious, whereas the last two would have more of a neutral effect. So that's about all that I have. So here are links to basically all of the NCBI resources that I was talking about, as well as our GitHub site. And now if anyone has any questions, we can get into that. And I think Ben's going to come back over and help answer those.
Does anybody have any questions? Somebody asked what was the criteria for choosing these programs. So we use programs that are fairly popular in terms of how much play they get on various internet forums that work with NCBI tools. That said, we don't endorse any tools, nor are we exclusive. So if you have other tools, please let us know, and we'd be happy to incorporate them into a future webinar of this kind. These webinars have been relatively popular, so and we really, in my opinion, NCBI should be very committed to making databases that work with external tools. Somebody else asked for variant impacts on 3D structure. That's something we're actively working on in the development space, and once we have sort of more stuff done, we have some new viewers that we've been releasing, like IC in 3D. And so once we have more variants reconciled with those 3D viewers, we'll probably be putting together a webinar on that. My guess would be early fall. Somebody asked about the program Gemini. That's potentially something we can add to a future webinar. Somebody else asked about Anavar's version of RefSeq and whether it's directly from NCBI or whether they modified it in any way. We will double check. I don't think it's modified, but we'll double check and we can get back to you on that. This webinar has been recorded to view later on. Once it's closed captioned, we will release it, and you can find that on the NCBI webinars page. One thing we can't answer is questions like what is the best program for X, Y, or Z. Once again, we can't endorse any of these community programs, but it is great that they all support using dbSNP data. With that, I think, unless there are any more burning questions, and like I said, some of the questions are relatively specific. We'll try to put some answers to those questions on our FTP site. And if you don't get an answer to your question, please feel free to send us an email, particularly if that question is about NCBI databases and not about the individual tools. Thank you very much, and I hope everyone has a fantastic rest of the week. And thanks again to Caitlin Williams for putting all this together.